We are honored to kick off the rally with a quick research update from Dr. Annie Huang. At the end of last year, Hope4ATRT and Tali's Fund teamed up to support the incredible work of Dr. Huang at the Hospital for Sick Kids, Toronto. Dr. Annie is a pediatric neuro-oncologist and scientist with the Cell Biology Research Program in the Arthur and Sonia Labat Brain Tumor Research Center at Sick Kids Hospital in Toronto. She established the Global Collaborative Rare Brain Tumor Consortium that has played an important role in advancing clinical and biological understanding of cancers like ATRT. Dr. Huang, thank you so much for joining us to share an update on this project.
Hello, everyone. Thank you so much for the opportunity to speak to families and thank you for having me and for supporting our efforts towards ATRT. I will give you a brief overview of the project that has been supported by Hope4ATRT. We're not by any means completed the project yet, but I hope it will help you understand where we're headed. So thank you again. Many of you know that ATRT is a rare kind of brain tumor, and that, even though we think of medulloblastoma as a very common tumor we talk about all the time, 40% of tumors that are like medulloblastoma, called embryonal brain tumors, are actually rare types of brain tumors, amongst which is ATRT and other tumors that we studied like ETMRs and pineoblastoma. What is very distinctive about this group of patients is that they're extremely young, as you all know, which is quite different, which limits what we can do medically in terms of trying to be aggressive with the treatment. So I need not remind people what ATRT is. It's a highly aggressive disease that affects very young children. What we call the median age is about 1.5 years. So they're really, and they can all be a lot less than 1.5 years. And about 20 to 30% of patients will have inherited the disease through a gene that they get. So historically, in the old days, when these trials were done with regular chemotherapy, the survival was very bad. So this red curve just shows you less than 10% of patients survived. We now know that, from a very important perspective, Children's Oncology Group trial that was run by Dr. Lisa Reddy from UCSF, that if you use what we call maximum intensity therapy, which is you use high-dose chemotherapy combined with radiation, the survival is better. So compared to children a long time ago that did not get high-dose chemotherapy, only 13% of those children survived beyond 4 years. Well, with this sort of intensive therapy, 40% of children survive beyond 4 years. What's important to note is that there's still a large proportion of children who are doing poorly very early on in this therapy, and that all of these children got everything we could give them. And so, this begs the question, what else can we do without causing a lot of different problems? So a question that remains outstanding in our field is, does everybody need this intensity of treatment? Because clearly there are survivors. Are those survivors surviving because of the high dose or because we've had improvement in technology for lots of different things? And is radiation absolutely necessary for all the children? Even if you give limited field radiation in a very small brain, you have the possibility of harming the growing brain, the normal growing brain. So at Sick Kids, we've always followed this paradigm that we tailor the treatment as precisely as possible to the child, given what the child is presenting with, how they're doing. And we try to cure with minimal toxicity. As these children are very young, and as they grow to be older, they have a lot of sort of struggles with their cognition, hearing, organ impairment, and all of those sorts of things. So the idea is that we cure children so they can continue to grow the way they're supposed to grow before their disease and go to do all the things that we hope our children will do. Go to school, have vocations, get married, have children, that kind of stuff. So with that aspirations, the Canadian community has banded together quite a while ago to think about doing something without radiation. That's the first thing we can try and get away with, because there was some really old literature that suggested that not every child who survived needed radiation. So our goal was to postpone as far as we can until the child is older and the brain is more mature, or never to use radiation if we didn't have to. So we largely followed the paradigm of the high-dose chemotherapy that has been used, but the drugs we use are a little bit different. It's less intensive than some of the drugs that has been used, with the thought that if we had to go to radiation, we would do less harm. So this is some of the data that's overlapped with the current data that showed that compared to children who received standard chemotherapy in the past, this group of children who received high-dose chemotherapy, a significant proportion of who never got radiation, was having a reasonably good survival, comparable to chemotherapy regimens that gave radiation as a mandatory part of the treatment. So, you know, this sort of brings home the point is of the children who survive, is there still a proportion that absolutely we can do away with lessen or extend their survival without causing a lot of harm? And so this is a child that I treated in 2007, CJ, many of you might have heard of him. He was 13 months old when he presented to us. We gave him the therapy we knew how to give, which was the high-dose chemotherapy. And at the end of the 6 months, CJ's spinal fluid showed a lot of tumor cells. And the options presented to the parents at that time was to give brain and spine radiation, but he was only 19 months old. This would have really hurt him. Or to try to extend his life without radiation, but with no hope for curing him. CJ is a very persistent mother that convinced us to attempt to do something different. And CJ was the first child in which we used CSF-directed therapy to try and get rid of these cells in his spinal fluid. And to be honest, I never expected him to survive. I expected to extend his life. But CJ beat the odds, and he is now 18 years old, actually, and has never seen radiation. He drives a car. He's trying to be an electrician. He has girlfriends. He's leading a normal life. So that is our aspiration for every child. And so we have persisted with this sort of approach, mainly because, not because we think that it can cure everybody, but to begin to learn how to tweak it so that we understand what gives and what doesn't give. The problem in the ATRT field is that we don't really know what the best approach is. And there are many different approaches. And so sometimes when you compare apples to oranges, it's really hard to figure out which one is a little bit different. So our approach has been to try and use the consistent approach so that we can begin to tweak it a bit more. And so this is just the beginning of the journey. So since that period of time, we've added 12 months of treatment with intrathecal chemotherapy to all the children who get high-dose chemotherapy. And here we never radiate until the tumor persists or it comes back. We added the CSF-directed therapy, which gave us an opportunity at 3 times during the beginning and 12 times later to sample the spinal fluid, to begin to think about how can we measure disease that we can't see. This is not earth-shattering because this has been done for leukemia. So when we started with CJ in 2005, we knew the technology will catch up. We just had to save the CSF. We tried many different things that did not work with the CSF. They couldn't, but we knew that this would be worthwhile to do. So we've been collecting this for a long time. We extended the treatment by 12 months, low-dose chemotherapy, just given orally regimen. The question always remains, is everybody needs all of this? And of the children who don't do well on this, could we have told earlier that we were on the wrong track? And how are we going to know without some sort of measure that is a little bit more sensitive than MRIs? So this is our results from 25 patients, treated over a period of 2005 to 2023. The blue chart shows you the patients who did not receive the 12 months of extra therapy. The red bar shows you the children who received the extra dose of chemotherapy. In these children, the majority of whom, 15 out of 25, has never received radiation. So there's a 60% survival. This is quite promising, as you know, the survival in other regimens that use chemotherapy is actually less than that. Use radiation is actually less than this. So we're encouraged by this, that the addition of an extended therapy, actually with CSF-directed therapy, has some merit. But how do we move forward on this now that we have treated 25 children like this? So how do we improve on this survival? 60% is still not good enough. We now know that ATRT is not a single disease, right? Even though there's only one gene mostly that's lost, we know that there's at least 3 subgroups of what we call subgroups of molecular disease. Presently, we don't really know what this means or how to tailor the treatment to the nuance of these groups. What we do know is that if in the laboratory, if you use cells that belong to each of these different groups, they actually respond to different types of drugs. But in practice, we actually do not know when to introduce the new drugs. Who do we give it to and when should we give it to them? That's because we don't have an established backbone. We don't have a routine way of knowing this is the best framework to treat these patients with. And so that was part of the reason why we went through this process since 2005 to figure out what we think would be the best framework for testing new drugs and making sure that we don't have to keep testing in many different shapes or form, but that we would hopefully arrive at an answer without too much effort. So we created this platform, which we call the C17 Baby Plate platform, which is a platform that runs across Canada. And the idea is that we would aim to create a very informed way of using the model that I showed you, where at the beginning of treatment, we collect a lot of material to find out more about the molecular type of ATRT that is, and start to think about a specific therapy that might be tailored to that. And then in all the children that follow this, we are measuring their CSF to see at different stages early on in the treatment and during the prolonged maintenance to see if we see any evidence of disease. And if there's evidence of disease, whether there was something that could actually help us decide how to change treatment. So we were lucky when, Jamie and Kim Doran got together and started discussing the projects we had been doing and the collection that we've had all of this time. And they very generously offered to support this effort with funds from Hope4ATRT and Tali's fund. And the idea is that we would use the CSF that has been collected from these children, and they are processed through a process like this, and we what you call gene sequence them or do the latest technology to look for very, very small amounts of DNA that is shed by the tumor cells if they are there. This technology is exquisitely sensitive, and it needed to be this kind of technology because if there are tumor cells that are not seen by MRI, they're usually things that require very precise technology to catch just a few cells that are there. So since 2005, we've collected 105 of these samples from the patients we have treated. Our intent, as I said, was to try to predict molecular relapse, which means that long before an MRI changes, we would know something is wrong. And long before the sequential MRIs that you're always told is free of disease, we would be able to tell you with some reassurance that your child is out of the woods. And perhaps starting with that, we can then shrink the duration of treatment. We can take away parts of the treatment that we think are unnecessary for some and absolutely essential for others, or not working for yet another group of patients. Importantly, the most important thing we want to answer from our question from the cohort of patients that we have is, where did we miss those patients who could use early radiation? And who are the patients who receive radiation that perhaps didn't need it at all, based on our assessment of their MRI? And then for some patients, how long can we postpone their radiation? With CJ, we postponed it forever. We treated him for a long time. Perhaps we could have treated him for shorter. And if we had this test when CJ was being treated, his mother would have allowed me to stop treating him for less than 3 years. So to date, we've analyzed 20 samples, and it's very encouraging because we are able to see tumor cells in some of the patients that we had not predicted would have relapse. And this is a work in progress. It's really detailed work. We hope the next time we speak to you, we'll have more to tell you about the results. But this is what we look towards. This is a slide borrowed from a review that was written about medulloblastoma. But this is what we want to do in ATRT, and this is what we had been visualizing and envisioning since CJ was treated. Very soon at the time of treatment is to take some CSF, which we do not normally do a bank or analyze at this time. And later on during treatment, we would continue to monitor this, and we would know when something is changing. So this paradigm is really not that different that has been used in leukemia for over half a century, I would say at least 50 years in leukemia, when leukemia patients always get the CSF tested to see if something is going wrong. We just have to do that catch up in brain tumors. So this paradigm has worked very well for some of the diseases, and I think will be very important for especially baby diseases, when we really do not want to give the 9 yards unless it's absolutely necessary. So we're now actually, based on some of this data, embarking on a clinical trial that we are trying to build across Canada. This is because our network is practicing the same way. Every child in Canada gets treated essentially the same way. But we did not have the funds to run a trial, and we had a patient group who again catched on like Kim Doran and Jamie, that we were maybe onto something different that they would like to help support. So we've just secured funds to run the first feasibility trial, meaning all the children in Canada are actually treated as such, but we never had the resources to collect the data except for the children at Sick Kids. But now that we will put them on a regimen and control the data and the way children are monitored and treated, we now will have a larger population to actually test these assays that we are developing and come up with a quicker answer. So this is very exciting for us, and we hope this will be launched in the coming year. And the first phase of it was to make sure that all of us are following the same rules and that we can get information reliably from our patients, every single patient across the nation. We're not a big nation, but we're not about to lose information on any child. So that's where we are. And I just wanted to thank everyone for your attention and for all the supporters that have enabled our studies. Our study started with Tali's fund supporting us with $35,000 of grant money. That was many years ago. And I can't thank them enough for believing that we can make a change. So thank you very much,
Dr.
for your
Huang.
attention.
Yes, thank you for sharing. We do have some questions coming in the chat. Unfortunately, because we do have some other featured speakers to get to, we don't have time for a Q&A with Dr. Annie, but I would love if you would just stay on and you could even answer those questions in the chat. And if anyone else has questions, feel free to drop them in the chat or email us and we can kind of continue that conversation. I know that was a lot to unpack. That was more than just an update. That was an incredible, decades worth of research there. And so we thank you so much, Dr. Huang, for just the work that you've done for ATRT and just the heart that you've put into it. We're excited to continue to, support this particular project and future projects as well. So thank you so much for sharing.